In this video, we're going to talk about fungi in the phylum Ascomycota, which are also known as the sac fungi. These fungi get their name due to the formation of a sac that contains haploid ascospores. This picture here shows you an example of a sac fungi. And this whole structure here, this kind of bowl-like structure, is known as an ascocarp. Now we can take a closer look at one of these ascocarps. If we look at this picture here, and this is showing you a cross section and also kind of zooming in to show the microscopic structures that make up this fungus. And you can see within this mushroom, there are these sac-like structures here. And each one of this is known as an ascus, which is singular, or asci, which is plural. And you can see that here with these arrows pointing to multiple asci. Within each of these asci, you can see the ascospores. So these little dots kind of inside here are the ascospores. The hyphae, which remember the hyphae make up the kind of vegetative underground portion of the fungal organism, and the hyphae in Ascomycota are septate, so they're going to look like the one in this picture here on the left. Remember the difference here is that the hyphae are made up of individual cells, and each cell has a nucleus. If it were aseptate, which would be the one on the right here, you can see that the cells are not divided by a cell wall because they're not really cells. It's actually just one big cell with multiple nuclei in it. Ascomycota fungi can do asexual and sexual reproduction. With asexual reproduction, kinetiophores release haploid kinetiospores. Kinetiophores are structures that extend from the hyphae of the fungal organism. You can see on this microscopic picture here that this whole structure here would be the kinetiophore. And the kinetiospores would be located inside, and then they would ultimately be released out into the environment, where they can then germinate if they find a suitable habitat. I want to point out the asexual reproduction that we talked about on the previous slide because you can see it on the diagram here. So remember that during asexual reproduction in Ascomycota, the mycelium, which is made up of the hyphae, forms the kinetiophore, which then it contains and then releases the kinetiospores. And so it's going to release these asexual spores called kinetiospores. And then after those spores are released, if they find a suitable place to land, they will go through germination, which is just mitosis, in order to form a new mycelium. For sexual reproduction, in Ascomycota, they have special hyphae that can develop into the different mating types. The kind of what we would call the male mating type is known as the antheridium, and the female mating type would be the ascogonium. Ascomycota can be homothallic or heterothallic. And what this means with homo, it just means that the antheridium and the ascogonium are going to be associated with the same organism. Hetero means different or other, and so in a heterothallic situation, the antheridium and the ascogonium are going to be associated with different fungal organisms. The Ascomycota go through the same basic steps of all fungal sexual reproduction. So the first step is going to be plasmagamy. Remember that plasmagamy stands for cytoplasm marriage. So during plasmagamy, we're going to have a cell from the antheridium fuse with a cell from the ascogonium. And we'll say the cell with the red nucleus is from the antheridium, and the cell with the blue nucleus is from the ascogonium. Remember that these nuclei are haploid, or 1N. And when plasmagamy happens, the cell walls, cell membrane, and cytoplasm are going to fuse together to form one cell. But the nuclei are not going to fuse together yet. And this forms a structure known as a dikaryon, which contains, it's a single cell that contains two haploid nuclei. And we denote this as N plus N. And you can see the structure on the diagram here. So you have the two cell mating types joining together during plasmagamy. The second step of sexual reproduction in fungi is known as karyogamy. And during karyogamy, that dikaryon that contained the two haploid nuclei, the nuclei are now going to fuse to form a cell with one diploid nucleus. So remember that this was the dikaryon formed during plasmagamy on the previous slide that had the two haploid nuclei, which we denoted as N plus N. And then the structure that is formed during karyogamy is a zygote. And it has a diploid nucleus that we denote as 2N. And I colored in this diploid nucleus purple because purple is what the color you get when you mix blue with red. But remember that in a diploid nucleus, you're actually going to have a combination of these paired red and blue chromosomes. After the zygote is formed, the next step is going to be meiosis. Now remember in meiosis, this is a form of cell division where the cell is going to go from diploid, or 2N, to form daughter cells that are haploid, or 1N. The daughter cells produced are also going to be genetically different from each other. They're also going to be genetically different from the original cells that they were formed from. Interestingly, Ascomycota actually have an extra step in their sexual reproduction cycle. So after the zygote goes through meiosis, during meiosis you have one cell that produces 4 daughter cells. That ascus then goes through mitosis and cell division, so those 4 cells actually become 8 ascospores. The ascospores are then released from the asci into the environment, and then if they land in a suitable environment, they will germinate in order to form a new phallus.